Hello, we are Group 8. I'm Georgia Zeloza, and our group members include Santiago Fernandez, Nicole Slusher, and Kaylin Vilas. So our topic is chemicals and fine particles in the air and soil. With this presentation, we aim to discuss the various types of chemicals and fine particles that are found in air and soil and connect them to cancer development in humans. Many of these substances contribute to pollution of air and soil, and it is through such pollution that humans can gain exposure to them. So an important aspect of substances causing cancer is determining if they are carcinogenic. A carcinogenic substance refers to a substance that's either a chemical, particle, or possibly a virus that has been linked to promoting cancer formation and proliferation. In the context of our presentation, we will be looking specifically at chemicals and fine particles as potential carcinogens. Carcinogens are linked to the promotion of cancer since they lead to an increase in DNA mutations. DNA mutations affect gene expression as well as have important implications for how various pathways are affected, and thus cancer promotion is possible. Carcinogens can be categorized into 4 groups, as seen on the slide. Group 1 refers to substances that are confirmed to be carcinogenic to cancer and thus are linked to the formation of cancers. Group 2 handles substances that are probably carcinogenic to humans, and Group 3 are those which are possibly carcinogenic. Finally, Group 4 refers to substances that are not classifiable as carcinogens. As we discuss chemicals and fine particles in the air and soil further, we will discuss specific substances, and many of these are classified as Group 1 carcinogens and are linked to cancer development in humans. Before discussing the various substances related to this topic further, it's important to understand how exposure occurs and how this exposure can lead to cancer formation. We will focus specifically on the air and soil when looking at potential exposures. For the air, exposure mainly occurs via inhalation of a chemical or fine particle. Depending on the size of the particle inhaled, it can travel to the upper respiratory tract or even further if it's a smaller particle. These substances can be inhaled from one's surrounding air, the environment, handling a material or item that releases these particles or chemicals into the air as well. Some of the most prominent reasons for these substances to be in the air come from factories, transportation, such as cars, trains, the use of aerosols, such as pesticides. In fact, some studies have shown that there's an increase in health issues related to air pollution when comparing present day to a time before factories. Such studies claim that there used to be fewer factories and thus less air pollution was being released from these factories. But as more and more factories have been built, there has been an increase in the particles and chemicals that they release into the air and thus increased exposure to humans. This has led to an increase to the exposure of such substances and an increase in health issues, including cancer developments. Exposure via the soil is a little less direct than air exposure. Many of the substances linked to the soil are metals or chemicals found in or added to soil via pollution. When one plants crops or vegetations in the soil, the substances found in the soil, such as these metals, chemicals, or particles, can then be taken up by the plants growing in the soil. This is because plants require soil for vital nutrients, but many crops are also sprayed with pesticides, for example, or planted in areas where the soil might be contaminated via pollution, such as runoff filled with chemicals. Once these plants grow in such soil, they are also now contaminated with these substances, and humans ingest them when they eat the plants produced. This is one of the major means of exposure to humans via soil, especially for carcinogenic metals. There's also greater exposure linked to various types of occupations. Farmers who work in fields and handle plants and soil have greater exposure to contaminated soil, but also to air pollution via pesticide use. Mechanics, or those who work with transportation, have greater exposure to gas and other inhalants that are linked to negative health effects. Construction workers, or those that work with house materials or building materials, face higher exposure to substances such as asbestos. Thus, one's occupation can be a factor that impacts their exposure to carcinogenic substances. However, even those that do not work in jobs that place them at a higher risk for exposure can still come in contact with substances in their daily lives, such as air pollution exposing individuals to dioxins or polycyclic aromatic hydrocarbons or PAHs, and other airborne inhalants linked to cancer. The use of cars or other forms of transportation expose users to diesel, gasoline, but much lower levels than the workers that work with these transportations. Exposure is also linked to the risk of cancer development, but many other factors affect this risk. Location can affect exposure, as observed by increased exposure to vinyl chloride in individuals living near plastic factories. And the risk can depend on the exposure type, such as how long one was exposed to a substance and how much of the substance they were exposed to. And risk is also linked to pre-existing health conditions and lifestyle choices, such as smoking. So throughout our presentation, we will look at specific examples of chemicals and fine particles, especially those which are carcinogenic. For those related to the air, we will look at diesel exhaust particles, polycyclic aromatic hydrocarbons or PAHs, solvents such as benzene, fibers, fine particles and dust, metals such as arsenic and beryllium, dioxins, and pesticides. For soil, we will examine pesticides, polycyclic aromatic hydrocarbons, dioxins, and metals such as cadmium, chromium, and nickel. So throughout this presentation, we will discuss current fields of knowledge of both the air and soil and examine how these various substances exist and spread in the environment, as well as how they're linked to exposure to humans. We will also discuss significant studies related to chemicals and fine particles in the air and soil. For particles in the air, we are going to examine a study relating to microparticles with a focus on asbestos and their relation to microRNA. And for particles and chemicals in the soils, we'll look at a source that provides detail on the various substances that could be found in soil, as well as how they've accumulated in the soil and how they could be exposed to humans in a variety of ways.
As you can see, there are many sources of pollution and chemicals in the air. They mostly arise from the combustion of fuels for various purposes, including energy industries, manufacturing, transportation, and industrial processes. Of particular interest are particulate matter and sulfur dioxide. Particulate matter, here labeled as PM2.5, is a percentage of each of these sources of pollution, between 3.6% for non-road transport and 43.1% for residential and small-scale combustion. This is also true for sulfur dioxide, with a range of 0.7% for road transport to 37.3% for energy industries. This accounts for much of the air pollution encountered outside of the home. Inside the home, there are two carcinogen sources of interest, asbestos and radon. Particulate matter is usually referred to as PM2.5, which defines the size of the particulates as less than 2.5 micrometers. While air pollution has been a health concern since the 1940s, air pollution and particulate matter were not classified as carcinogens until 2013, when they were placed in Group 1. Additionally, the EPA recently revised the regulatory limits of PM2.5 from 15 micrograms per cubic meter to 12 micrograms per cubic meter in 2012, in response to data supporting the detrimental health effects of air pollution. A cohort study from 2002 looked at data collected since 1982 and an assessment in 1998 that included 500,000 participants. It found that for every 10 microgram increase in particulate concentration, there was an 8% increase in the risk of lung cancer mortality. This study controlled for tobacco smoking status and found a strong correlation for smokers and an even higher correlation for people who never smoked. Here you can see that the mean for smokers was 1.04 relative risk of mortality, and that for people that never smoked, it was 1.2 or 1.12 compared to an unexposed group. Another large contributor to carcinogenesis of air pollution is sulfur dioxide. In the form of SO2, sulfur dioxide, it is classified in Group 3, not classifiable as carcinogenic to humans. But when it is oxidized into ammonium sulfate by interactions with other constituents of acid mist, what we would commonly call pollution fog or haze, it is classified as Group 1. High levels of sulfate aerosols have been shown to promote lung cancer development. Oxidized species can activate EMT transition and promote metastasis through E-cadherin promoter methylation. Here you can see a chart with results of a study that showed how ammonium sulfate increases metastasis of lung cancer compared to an inert vehicle. Inside the home, there are several carcinogenic sources that can be encountered, particularly radon and asbestos. Radon is a naturally occurring element formed from the decay of radium and is classified as a Group 1 carcinogen. It was first identified as an indoor health concern in the 1980s by a power plant worker who discovered it accidentally. He had carried a radiation detector into a power plant that had not yet received radioactive materials, and the detector was picking up a concentration of radiation. The concentration of radon is measured in a unit called becquerels per cubic meter, or BQ over meter cubed. It has been found that the risk of lung cancer increases by more than 8% for every 100 becquerel per cubic meter increase in concentration, with an even higher percentage for smokers. For example, you can see in the chart here that for the percent mortality from lung cancer for a non-smoker with a radon exposure of 200 to 399 becquerels was 0.47%, and for a smoker with the same level of exposure, the percent mortality from lung cancer was 11.6%. Asbestos is another naturally occurring genotoxin and carcinogen. It is a fibrous mineral that was used widely for its corrosion and heat-resistant properties. The earliest found study linking asbestos exposure and adverse health effects was published in 1955. This was a follow-up study for a 1935 study of asbestosis in asbestos workers. Circumstance of death information was gathered for the workers from the previous study, and a significant higher than normal incidence of lung cancer was found. Before the 1990s, when production of asbestos-containing construction materials was largely banned, asbestos was found in ceiling and floor tiles, insulation, shingles, textured paint, and heat-resistant materials around stoves and pipes. The currently allowed commercial uses for asbestos include brake pads, roofing materials, and vinyl tile. So while it's not common in new construction, it's especially important to keep in mind when doing home renovations on houses built before 1975. Exposure to asbestos is associated with a specific rare type of cancer called mesothelioma, which affects tissue lining the outside of the lungs. Exposure through inhalation primarily can cause chromosomal damage, often deletions and segregation errors, because it can interfere with mitotic spindle function. It can also cause reactive oxygen species formation and inflammation that can lead to fibrosis. We'll cover asbestos a little further later in the presentation. Two carcinogen sources of concern in both the air and soil are polycyclic aromatic hydrocarbons, or PHAs, and dioxins. PHAs are a group of hydrocarbon compounds that can be found in tobacco smoke, grilled meat, combustion products, of fossil fuels and wood, and many industrial processes. When they are aerosolized and inhaled, they have been linked to lung, bladder, and skin cancer. After aerosolization, they can be put in the soil as they settle or by rain, and here they can be sequestered into some plants, and they can also be directly ingested as contaminated soil on improperly washed produce. Ingestion of these compounds has been linked to bladder and gastrointestinal cancers. The earliest study about PHAs that was found was an epidemiological study in 1936 of workers in Japan and England who were in the coal carbonization industry, and they're presently classified as Group 1 carcinogens. Dioxins are also classified as Group 1 carcinogens, and like PAHs, they are usually encountered as byproducts of industrial processes and pose the greatest risk to those that work in the industry. There have been two notable industrial incidents where populations were exposed to dioxins at high levels for extended period of time: Love Canal and what is known as the Seveso incident. In 1976, in Seveso, Italy, an industrial accident exposed a large population to high levels of dioxin. In a 1996 follow-up to the incident, there was found a higher than normal incidence of lymphatic and hematopoietic neoplasms, and an increased risk of breast cancer for people who lived near the site. A further mortality study was done 15 years later and found increased mortality for digestive and stomach cancers in women and increased mortality for rectal cancer in men. Here you can see a proposed mechanism of action where dioxins bind to AHR, aryl hydrocarbon receptor, which when activated enters the nucleus and promotes transcription of cytochrome family enzymes that help metabolize dioxin, but also promote the formation of reactive oxygen species and can cause genotoxic effects. Turning our attention to soil pollution, you can see that, like air, there are many sources of contamination, either from natural or anthropogenic sources. Particulates and chemicals can be released into the air from combustion, wildfires, and transportation, and they eventually settle on the ground. But they can also be directly applied to the soil through pesticides and industrial chemical runoff. Two carcinogens worth noting are the naturally occurring heavy metals, arsenic and chromium. Arsenic is a naturally occurring metal and a group 1 carcinogen found in pesticides and as a byproduct of industrial processes. It is also used as an anti-cancer treatment for acute promyelocytic leukemia and in the treatment of malaria. Diet is the largest source of exposure, but it can also be introduced through the air, and the severity of the health effects depends on exposure time and quantity. It has been associated with higher mortality rates for cancers of the skin, bladder, liver, and kidney in areas with notable levels of arsenic contamination in the soil. This chart outlines different mechanisms of action for arsenic, which lead to many types of cellular disruptions, from decreased CD4 cell activity to chromosomal abnormalities and epigenetic changes that can lead to cancer development. Finally, chromium is an element present in several oxidation states, where chromium-3 is the natural state of the element and classified in group 3, while chromium-6 is a form classified as a group 1 carcinogen. Chromium-3 is an essential nutrient for human metabolism and potentiates the action of insulin in the body. And most exposure and negative health effects occur in industries that use chromium, like metal processing, leather tanning, and welding. Inhalation of chromium-6 and skin contact are linked to respiratory cancers. It can also contaminate soil, though it poses less of a risk through contact sensitization from those sources. On contact, it causes oxidative damage and has genotoxic effects.
Okay, so as previously mentioned, the susceptibility specific microparticles have to become inhaled and promote pathogenicity in the form of numerous illnesses and cancers, more importantly, is a lot more common than people would come to think about. One prime example of such microparticles, as we mentioned previously, is asbestos. So asbestos has a long history as one of, if not the most popular go-to insulation material found in the common household. This is due to the nature of asbestos' exceptional tensile and fiber strength and resistance to up to 1600 degrees Fahrenheit. So naturally, one can come to understand why it's such a good insulator. Multiple studies have observed the effect asbestos have within the realm of respiratory illnesses. And one such study, titled Role of MicroRNAs in Lung Carcinogenesis Induced by Asbestos by Bersimbaev et al. 2021, is going to be sort of the primary case study that I'm going to be referencing within the next couple of slides. And it covers the role of asbestos in promoting oncogenesis at the molecular level. So physiologically speaking, consistent inhalation of asbestos fibers can result in fibrosis of the mucous membranes that line our respiratory tract, as Nicole mentioned previously. Among the most common physiological responses, of course, is inflammation, which is a natural response that is linked to the increased susceptibility of multiple cancers. One such that we covered in class, such as colon cancer, which we learned is due to the irritants harbored by heavily processed foods here in the US. With all this being said, consequently, it becomes evident that chronic exposure to these asbestos microfibers really has the potential of increasing and inducing carcinogenesis. In recent studies, asbestos has been examined under the scope of promoting oncogenesis in the form of lung cancer, of course, by inducing reactive oxygen species, or ROS, into microRNAs. Now, what exactly are microRNAs? Well, microRNAs are essentially very small, typically around 22 to 24 nucleotides in length. Of course, some can vary either above or below that range. But more importantly, they are non-coding RNA snippets, and they serve to inhibit the expression of mRNA by cleaving certain sites of that messenger RNA, but they can additionally regulate gene expression at the post-transcriptional level. If we delve into the microRNA-dependent mechanisms through which asbestos fibers promote carcinogenesis, we need to, of course, note that most microRNAs play vital roles in modulating cell proliferation, differentiation, and apoptosis, or cell death. So the introduction of asbestos into the human respiratory tract has been observed as impacting the microRNAs that oversee cell cycle factors in the lungs. I've listed here, some of them include MIR103, MIR22, MIR20A, so on and so forth. Consequently, many mRNAs linked to cyclin-dependent kinases, for example, CDK4, CDK6, these are kinases that we covered in class, responsible for regulating stages of the cell cycle. They have been found to be either overexpressed or inhibited, depending on the concentration of inhaled asbestos microfibers in individuals. I also decided to include some relevant information on P53, which we also have covered in lecture. We have all seen recently that P53 is remarkably noted as the master tumor suppressor and modulator of cell death and apoptosis in the cell. And studies have demonstrated that there is, in fact, a correlation between the microRNA profiles that I have listed here at the bottom of the slide, such as those of MIR504, and the inhibited expressed levels of the TP53 gene, causing an overall hindrance of apoptosis and cell death. And of course, this is all influenced by asbestos microfibers, which, I mean, naturally one can see why it promotes oncogenesis. So for this next slide, we have two figures on the right, and I want to draw the attention, of course, to the utmost figure. As we all know, lung cancer remains one of the most fatal cancers, with an exceedingly high mortality rate in both men and women, as demonstrated by the first figure on this slide. Specifically, as Nicole covered briefly, the type of lung cancer induced by asbestos is referred to as mesothelioma, which is the formation of carcinogenic tissue in the mucous membranes enveloping not only the lungs, but parts of the thoracic cavity and the heart as well. So high levels of asbestos microfibers that reach deep inside our respiratory tract, aka the lungs, have been shown to enhance local genotoxic stress. And what I mean by genotoxic stress is essentially any environmental factor that can abruptly induce biochemical changes that elicit DNA damage. The DNA repair machinery that is present in the human genome is very complex, and it is disrupted easily by such biochemical alterations. For example, microRNA 138, a tumor suppressor shown to be overexpressed in carcinogenic lung tissue, hinders DNA repair due to a negative feedback response induced in the cell cycle. Moreover, other miRNAs, such as LET7D, LET7E, and MIR126, essentially function as tumor suppressors that diminish the susceptibility of developing lung cancer. So if we keep this in mind and analyze other studies where researchers have tested these miRNA levels, they have found that in certain individuals exposed to asbestos, these miRNAs, the tumor suppressors, are recorded in much higher levels. So if we connect the dots, we can understand that if in patients or in individuals where asbestos concentrations are high and tumor suppressor miRNAs are exceedingly high when compared to the wild type or the control group, one can connect the fact that asbestos does in fact play a role in carcinogenic formation. In the figure below, figure 1, we have sort of a diagram provided by the case study I mentioned earlier, which sort of classifies some asbestos-related microRNAs, a few of which I've mentioned, oncosuppressors microRNA, the ones I just spoke about, and oncomyrs in lung cancer as well. Next slide, please. Thank you. So overall, the correlation between miRNAs and asbestos and the prevalence of these two factors in cancer, specifically, as we mentioned, mesothelioma, it's no question that their relationship has been extensively proven. Domestic mining for asbestos here in the US was finalized in 2002. But of course, there has been many cases of importing the material. So albeit it's not mined anymore here in the US, it of course is still very prevalent in our society. As we spoke briefly prior, the major groups at risk of developing lung cancer from asbestos exposure naturally include those working consistently with insulation materials, and not only those, but also textured paint. By textured paint, I'm referring to a sort of paint that kind of brings out a 3D image in a wall, or it makes the wall look three-dimensional. Again, very common in older home styles. And of course, it's advisable for people who work around these factors to wear protective respiratory gear and clothing to avoid inhalation or exposure to these microfibers. But another interesting thing to note, aside from preventative measures, is some of the future directions this extensive research on microRNAs can lead, more so in the clinical setting. So I spoke previously as to how some microRNAs, particularly those tumor suppressors, are expressed a lot more highly or in higher levels in patients who have been exposed to asbestos. And we can essentially use these miRNAs, for example, I have here MIR155, as potential bioindicators for lung cancer. This was a pilot study that was tested in mice. Sorry, not a pilot study. This refers back to the original case study I'm referencing. But they tested the levels of MIR155 in mice who had been, of course, exposed to asbestos. And they found that these miRNAs were expressed in much higher levels in mice with the lung fibrosis. So this goes to say that perhaps a diagnostic method to start identifying variations of mesothelioma and lung cancer in patients could be to perhaps reimagine ways of scanning for particular miRNAs.
So we will now look at a significant source regarding soil. So a multitude of organic and inorganic materials plays a crucial role in supporting life on Earth and making soil a complex system. Nutrients, gases, minerals, microbes, and chemical substances all coexist in the world. However, not all soil-derived chemicals are beneficial. As previously mentioned, some have been linked to human cancer development, making them harmful. There are a few classifications of soil that can be connected to malignant growth. Heavy metals, pesticides, polycyclic aromatic hydrocarbons or PAHs, and volatile organic compounds are the primary categories. Natural elements with a high atomic weight and density are considered heavy metals. Arsenic, cadmium, lead, and mercury are all commonly considered heavy metals found within the soil. These metals are found naturally in the environment, but human activities like mining, agriculture, and various industries can lead to increased concentrations. Cancer, among other illnesses, can result from prolonged exposure to these metals. For instance, arsenic exposure has been related to skin, lung, and bladder cancers and diseases. Mercury can harm the nervous system, and cadmium has been linked to cancers of the prostate and lungs. Chemicals such as pesticides are used in agriculture to get rid of pests from the crops. Organophosphates, organochlorines, and carbamates are among the carcinogenic substances found in many pesticides. Pesticides can build up in the soil, leak into the groundwater, and make their way into the food chain via plants or contaminated water. Cancers like leukemia, non-Hodgkin lymphoma, brain, kidney, and prostate cancer may be linked to pesticide exposure according to epidemiological studies.
PAHs are a gathering of more than 100 unique synthetics that are formed during the inadequate consuming of coal, oil, gas, wood, trash, and other natural substances. They have the ability to stick to soil particles and remain for years. Cancers of the bladder, lungs, and skin have all been linked to the exposure of PAHs. At room temperature, VOCs are organic chemicals with high vapor pressure. They can be set free from consuming fuel like gas, wood, coal, or from solvents, paints, pastes, and different items. Vinyl chloride, formaldehyde, and benzene, all of which have been linked to leukemia and lymphoma, as well as lung cancer and liver cancer, are examples of VOCs that are carcinogenic. There are a number of possible routes that soil chemicals and cancer can take. Ingestion is a significant route. Children's actions, like hand-to-mouth play, put them at particularly high risk. Additionally, certain soil chemicals can be absorbed by plants, which can then enter the human food chain. Another way is to inhale soil or dust particles. Additionally, some chemicals have the potential to enter groundwater and contaminate sources of drinking water. Moreover, soil bacteria have the ability to transform some of the soil's carcinogenic compounds into even more harmful ones. Nitrosamines, which are potent carcinogens, can be made from common agricultural chemical nitrate This biotransformation process might make some chemicals in the soil more likely to cause cancer.
So nonetheless, it is essential really to understand and fully comprehend that the simple presence of these synthetic substances doesn't naturally compare or corroborate to disease risk. The portion, span of openness, the singular's well-being, and genetically hereditary factors all have impact in deciding the gamble of what this exposure truly means. To reduce the dangers posed by soil contamination, there has to be some form of method prevention and additional controls that have to be taken. These include better waste management, stricter regulation of industrial emissions, more sustainable farming techniques and practices, all of which can lead to cutting down pesticide use and ongoing monitoring of the quality of soil and water. To get rid of harmful substances from contaminated soils, a remediation process like phytoremediation and bioremediation can be enacted. With this, although soil does play a crucial role in Earth's ecosystem, it can store chemicals that can lead to an increased cause of cancer. To fully comprehend the intricate connections between soil contamination, human exposure pathways, and cancer risk, there needs to be more extensive research to back up these claims, essentially. Meanwhile, precaution and mitigatory measures can assist with safeguarding human well-being and, of course, the climate.
So throughout this presentation, we've aimed to look at the connection between chemicals, fine particles in the air and soil, and formation of cancer in humans. In doing so, we have identified substances that classify as carcinogenic to humans and to which they face exposure via air and soil. We examine particulate matter and the role it plays in spreading chemicals via the air, as well as the increase in particulate matter and its classification as a carcinogen in recent years. It is important to note that many of these carcinogens in the air are specifically linked to increases in lung cancer. It's also important to note that the air was sulfur, which was mentioned, and is not always carcinogenic, but can be based on the form it takes. We examine other forms of air pollution that could be found in homes as well, including radon and asbestos, both of which are carcinogenic and once again have ties to the development of lung cancers. There are some chemicals and fine particles that could be found in both the air and soil, such as PAHs and dioxins. PAHs are linked to a variety of cancers and can be found in the air via burning materials, industrial processes, and in the soil, they lead to exposure when humans consume crops grown in contaminated soil. Dioxins function similarly to PAHs in their means and exposure via air and soil, and they're linked to a variety of cancers. In the soil, we examine pesticides and various metals such as arsenic, chromium, and both of which are considered carcinogenic. We also looked to specific studies that we felt were significant to our topic. For the air, we examined a study that looked at microparticles in the air, specifically at asbestos and its resulting effects as a carcinogen. Through this study, it was determined that asbestos exposure plays an important role in promoting oncogenesis via affecting microRNAs, and increased exposure increases risk of such a development. There are also links to the specific effects these carcinogens cause in certain pathways, such as impacting the expression of the TP53 gene. There's promising research regarding the potential use of microRNAs as an indicator for cancer. Regarding the study for soil, we looked further into how chemicals can be found in the soil and lead to exposure in humans. In doing so, we see that consumption of crops grown in contaminated soil is a major source of exposure, but that there is also exposure via contact with soil, especially amongst children when they spread it via play or with each other after exposure to soil. We've also investigated means of prevention for the spread of these contaminants. For both air and soil, frequent quality checks should be implemented to provide people with warnings of potential exposure to carcinogenic substances, as well as to determine which areas face higher exposure than others. Managing wastewater, overuse of pesticides, or contaminated runoff can help prevent the leak of chemicals into the soil, especially to crops. Limiting air pollution and the use of aerosols, as well as other carcinogenic materials, such as asbestos, can further aid in lowering the amount of people exposed to carcinogens. Also, utilizing protective respiratory gear can further help lower exposure to airborne chemicals and fine particles. Thank you so much for watching our presentation, and please leave any questions or comments in the discussion.